My name is Dean Petkanas. I'm the Chief Executive Officer at Kannalife Sciences. We always focused on cannabidiol. We never focused on any of the other cannabinoids that are out there, and some may have some real interesting pharmaceutical value, but we really zeroed in on CBD primarily, and the use of that in a neurodegenerative, neuroprotective manner. So we had to focus on the areas where we felt that it would be best utilized. And that's where we began our research and continue to do so. We're probably within a year of doing a clinical protocol and design for a study. We'd like to open up that study in Europe. We think Europe is a good market in which to open up that study for the use of CBD as a preventative. We think that CBD is much an omega-3, a super omega-3 is a fish oil, which has largely been used for aches, pains, cartilage repair. Even some take fish oil for healthy brain function. And when you look at the compound structures in terms of omega-3 complexes, you see that CBD might have a strong position in the marketplace, maybe more so than some of the other substrates out there like a fish oil. We set up a set of rules from day one. Now, we had other potential business ops that were designed underneath the Kannalife umbrella. But from the pharmaceutical side, we stridently focused on CBD, focused on the HE model, focused on orphan designation and neuroprotection. And our preclinical work in that area confirmed where we should continue going. Because many times companies go into preclinical with target drug candidates and they don't prove out. And you just say, okay, we gave it a shot and it just didn't show up. And that's not the case. So from our original point of concept, when we were conceived as a company and the rules were set in place of what we wanted to do, we've accomplished those objectives and we continue to move towards that pathway. We looked at Cannabidiol as a lead target drug candidate for the hepatic encephalopathy model and for the CTE model. But we knew from early preclinical research that there were limitations to cannabidiol in terms of bioavailability, permeability. It's not an aqueous compound. It's a very oily, greasy compound. And so there are limitations in terms of quantity, dose control, also residency, half-life. And I looked at the possibility that there are other plant sources out there, inclusive of the design of a synthetic model, which we did with 019 and other similar cannabidiol-derived compounds, that there's some other plant sources that were related to cannabis where there might be a potential for a pharmaceutical ingredient within those plants that would be a high neuroprotectant, like CBD. So we found some very interesting properties in hops. And now we've pretty much done as much research as we could do on hops, and we think that we've gotten to a point where we're close to filing some patent work on that. So we wanted to have a bracket around CBD so that we have some options to work with in terms of the dynamic of neurodegenerative related diseases and treatment thereof, that CBD has some competitive field that we created within the spectrum of cannabinoid therapeutics, and not waiting for outsiders and competitors to do that. So we tried to be very forward-thinking and dynamic and lateral in our approach to neuroprotection.